Hello everyone, I'm Anushua Biswas, and before I present today, I would like to thank the RSG DREAM organizers for allowing me to present my work here and giving me this opportunity. So I would go ahead and begin. I would be talking today on identifying different modes of TF-DNA interactions in ChIP-exo datasets. Now, ChIP is chromatin immunoprecipitation is one of the most common methods to identify the protein binding sites in the DNA. It brings down all the DNA regions that are associated with the target protein, be it a direct or an indirect binding site. When these regions are mapped to the genome, then we see peaks forming, which are like peaks of these DNA reads, which form at the positive and the negative strands. Somewhere in between these peaks, the protein binds. So to find out the exact location of the protein binding, we do motif discovery in these peak regions. Okay, we note here that a single overrepresented motif may not be able to explain all the regions pulled down in the ChIP experiment. A protein can cause immunoprecipitation because of many reasons. For example, over here, the red pentagon protein, which is being profiled here, is bringing down different regions because of the different modes of binding. Our earlier method, diversity, addressed this question, and it divided the dataset into different modes of protein binding based on just the sequence information, while also identifying the corresponding motifs. Now, with advances in NGS technologies, we have a modified version of ChIP-seq, which is ChIP-exo. It maps the TF binding sites, but with a higher resolution. The initial steps of this process are similar to that of ChIP-seq protocol. The proteins are cross-linked to DNA, then the DNA is sheared, and then the target proteins bound to the DNA regions are immunoprecipitated using antibodies. These immunoprecipitated DNA are then adapter ligated and treated with exonuclease enzyme. Exonuclease enzyme digests the free DNA from 5' to 3' end and till it encounters the bound protein. At this stage, the crosslinks are reversed and the DNA is sequenced and mapped to the genome. So because of the exonuclease digestion, we see that these DNA reads are mapped very close to the protein binding site, forming sharp and precise read distributions. So we look towards including the insights from our earlier method, diversity, to check whether using the reads along with the motifs would help us to better identify these protein binding regions. Other computational methods also use these read distributions to understand better the protein binding modes in the DNA. Okay, so existing methods are like exoprofiler by Starik et al. They use motifs which are known either from a motif database or from a de novo motif discovery algorithm results. And then they scan these peak regions with these known motifs. For every motif, they calculate the read coverage on both the forward and the reverse strand, and then they plot the sequence heat map and the read profiles for every motif. A more advanced algorithm, ChEx-Mix, uses EM, and it divides the dataset into different modes binding subtypes. So these are done on the basis of both the read distribution and the motif information. But the initialization is key here. That's because it is done either by using the read distribution or by using the motif information, which can introduce a bias in the subtype estimation. So we present our framework, exodiversity, which is a probabilistic framework on both the read distribution and the sequence information, and it uses both of these to together learn the different modes of protein binding in the dataset. The input to this algorithm is actually in the form of 3 sets of sequences. The first set is a set of DNA sequences about the summit region. So this is denoted by X, and we can see a signal at the center, which corresponds to the summit. Now, corresponding to every sequence in X, we have the positive strand read counts RP. And here we see a signal to the left of the center. And similarly, we have the negative strand read counts sequences Rn, where we see a signal just to the right of the center. Now, the question here is basically, is there a single type of motif with its reads on both the strands, or are there different motifs with different read distributions corresponding to different modes? Now we should note here that the motifs may not always occur just at the center where we see the signal. So let us suppose that there are M modes in this dataset. We will build a model capital M with the parameters theta M. Consider the DNA sequence Xi, it will have the motif somewhere. Corresponding to Xi, we have the positive strand read counts, RPI. It will have the read window somewhere to the left, and we consider a 5 bases read window only. Similarly, we have RNI, which will be the negative strand read counts, and it has a read window of 5 bases somewhere to the right of the motif. Now, the parameter Zi would denote the start position of the motif in each of these DNA sequences Xi. Then we have II, which would denote the mode, binding mode for the sequence Xi. Then WK, which would be the width of the motif. And then we have these parameters tau. So tau pk is the relative distance between the motif and the positive strand read count window. The tau nk would be the relative distance between the motif and the negative strand read window. Then we have these phi's. These phi's are the matrices. So phi k is the categorical distribution over ACGT at the motif position for mode k. And phi pk and phi nk would be the Bernoulli read distributions, which would indicate the presence or absence of read in the read windows for positive and negative strand. All these parameters are unknown, and the goal is to learn the values of these parameters to maximize the posterior probability. Down here we have the posterior, which is proportional to the likelihood times the prior. So our model is actually based on collapsed Gibbs sampling. For each sequence, we first sample the motif start position and then we sample the mode. And this would help us to build the PWM or the phi k for each mode. Then we go on to sample the position of the positive and the negative strand read window. This would basically give us the values for tau, pk, tau, nk, and also define the read profiles for each mode. After that, we sample the width of the motif on both the left and the right side and update the PWM or the phi's. And this whole process is repeated till the likelihood of the whole data does not change anymore. We create models with varying number of modes, and then using Bayesian model selection, we choose the model with the maximum posterior probability. This is the input, which is basically ChIP-exo for CTCF and NCF7 cell line. So exodiversity was run on this, and it divided the whole data set into 16 modes of binding. This is the canonical CTCF motif in human. Now we see that these modes are somewhat similar in the motif signal, but they differ in the read distributions. So we wanted to investigate whether they are functional differences or they are just mere artifacts. So we look at the modes 1 to 7 first. We go ahead and see that the motifs are pretty much similar. But if we look at modes 1 and 2, we see that there is a band of high concentration of read counts. And this band is also there in mode 2, but they are shifted. Now, if we look at modes 2 and 3, we see the read distributions are very, very different. So we take a closer look at modes 1 and 2. And here we look at those positions where we see the band. Here, if you see, we have plotted the difference between the mean of the read counts for modes 1 and 2. And we see that they are either less than 0 or greater than 0. And when we looked at the distributions at these points, we saw that they are significantly different, which are indicated by the squares. Now we go to look at modes 2 and 3. So modes 2 and 3, if we see, the read distributions are anyway different. This is reflected here when we do a difference between the mean read counts between modes 2 and 3. All of them are greater than 0, and at each point the distributions were significantly different. If we plot the conservation plot here, these sequence conservation values, for modes 2 and 3 we see these also differ. So now if we look at modes 8 to 13, the rest of them we see for small changes in the motif, we have changes reflected in the read distributions on both the forward and the reverse strand, and also in the conservation scores. So we ran exodiversity on some other data sets. This is for FOXA1, where we again see the modes come up with very small differences, but they have differences in the read distributions. In the GR dataset, we saw that the only mode 3 corresponds to the canonical GR motif. All others are having different motifs which are of different TFs. they might be doing some cooperative binding with GR. Next, we see the summary. Finally, so our method learns a joint read distribution over reads and sequences and can identify distinct modes of binding. But they can be similar in one aspect, but different in other aspects. Our method does not clearly look for overrepresented motifs in the dataset, and it also does not assume that all modes would have reads in the most enriched peak regions. So our method really looks for small read windows in contrast to others that look for broader read profiles. So it finds small differences in the motifs that reflect in the read profiles and also in the conservation profiles, which suggests that these might be functional differences and not just artifacts. To end my talk here, I would like to thank Sean Mahoney and Naomi Yamada for their help, their initial suggestion of looking at the ChIP-exo dataset using both the reads and the motif information, and later on their help with the datasets and understanding of their method. Then I would like to thank Rahul Siddharthan for discussions in the later sessions and also my funding agency. And if you have any queries, you can always contact us using the following emails. We will be putting up the code for exodiversity very soon on GitHub, so it would be available to everyone. Thank you.
All right. Hi, everybody. Great talk, Anushua. That was really interesting, very interesting method. I think these probabilistic models, a very interesting approach to model these kinds of high resolution data sets.
Yeah.
I really like the fact that you're able to pick up this interesting diversity in the footprints, and also able to validate it statistically based on the read distributions. I was just wondering, for your CTCF examples, did you see other kinds of co-motifs nearby which would explain the differences? Because you mostly just looked at the primary motif below the footprint, but what about motifs around the primary motif?
We haven't yet looked at motifs which are farther away from the summit. We had considered a 200 base pair window about the summit to look for the motifs. So to investigate these after the sequence conservation plot, we were going to look at what are the other factors which are contributing to these motifs being different and having such different read distributions. So one of the approach was to look at a broader motif. So in the CTCF motifs which we see, there is a very faint signal to the left as well as the right. So when we took a motif width as 50, then we captured those as well. So we are still looking into the flanking regions to find out some other differences which can explain why these motifs are different in very small proportion and still having different read distributions, how the exonuclease is working for these binding events.
Great. There are two other related questions. So one is from Julia Zeitlinger. She asks, how does a model perform when two motifs are close to each other? Which is, I guess, similar to what I was asking.
Yeah. So the thing is, when it is clustering, it is just looking for a single motif in a particular mode. So it will take up that motif which has a greater contribution mode-wise, the motif-wise as well as the read distribution-wise. So the other motifs which may be not that so prominent and may not be having a very good read distribution may get missed. It won't be captured.
Great. Melanie asks, how does a model perform when the ChIP-exo profiles form a broad distribution on both strands at a binding site?
Right. So that is not fixed. The five base read window is not fixed. We can change it. We can increase it to capture a broader read distribution, but we haven't checked beyond 10 bases. So since if the exonuclease, which is used in both the ChIP-exo and ChIP-nexus experiments, cut very close to the protein binding site, we assume that it may be over a very small region and not like ChIP-seq, where we have a broader read distribution.
Yeah, that's a good point. I think what has been often found is that when the TF binds indirectly, like it's tethered, you typically get broader profiles. And maybe that's one of the reasons the motifs you're picking up are usually the primary motif that the TF binds to. Yeah, I don't think you saw much evidence of secondary motifs of tethered binding to other motifs of other transcription factors. Do you see that?
Not in CTCF, but in the FOXA1 dataset, we do see that. We have
Okay.
CTCF motif come up in the FOXA1 dataset as well. So that is it's a reported event where they say that there is a tethered binding where FOXA1 is interacting indirectly through the CTCF thing. So we would like to try it out on more datasets and look at them. So and that would be great. We are looking at some other data sets as well to find more indirect binding proteins so that we can get more information out of that.
Excellent. So one last quick question. Did you compare your results to those of ChIP-exo? And if so, did you observe major differences? This question is from Irene Kepler.
Okay. So I did compare the results with ChIP-exo, and for CTCF now, ChIP-exo, so the best result was ChIP-exo was giving us around 6 modes, and they were also smaller variations in the modes. But ChIP-exo looks at a broader read region, so we do expect that they would be a little different from what we find, and there will be lesser number of subtypes, which was true. And for FOXA1 as well, we ran ChIP-exo on the dataset, and it again gave 2 subtypes, which were 2 variations of FOXA1. And for the GR data also, we ran ChIP-exo, and it gave pretty many variations. But so currently, ChIP-exo is good when we want to look at broader read regions, and we want to cluster based on just the primary motifs as well as the read distribution, or even if we don't have a motif. But our method is basically looking for a motif and read distribution combination. Both of them should be present. And so
Great.
That's how it's different.
All right. Thank you so much, Anushua. That was a great talk.